Hello everyone, welcome to VinAm Guidance. This is Dr. Anuja Sharma, and in this video we'll discuss some of the questions from regulation of gene expression in prokaryotes, lac operon, mutation in lac operon, TRP operon and general operon system. So let's get started. Now before we move forward in this video, let me share some important news with you. Unacademy brings to you Arise, the first batch for IIT JAM 2022 Biotechnology exam. This batch will start on 4th of January 2021, so it's time to enroll. In this course, top educators like Rabish Tiwari, Debashish Kar, Manish Pallavi and Chaparla Chandrasekhar will cover the entire syllabus of biotechnology. These classes will cover topics from general biology, molecular biology, basic biotechnology, biochemistry and physiology, cell biology, microbiology, and biotechniques. The batch currently has 2 courses and more courses will be added soon for the above topics as the batch progresses. Each course will also have a doubt clearing session to help you clear any doubts of any topics. The entire schedule is given on Unacademy website that you can access using the link below. This course will include strategy, tips and tricks, practice questions and time management techniques for IIT JAM exam. Enrolling into this batch will be the one-step solution to prepare and revise for IIT JAM exam. The courses will be covered in Hindi and the notes will be provided in English. I think you'll definitely benefit from this course despite the stage of your preparation and move one step closer to your target, that is to crack IIT JAM 2022. So I suggest you don't miss this great opportunity and subscribe soon. Click on the link in the description box and subscribe now. Use the referral code VinAmG when you subscribe and buy a plan on Unacademy. You can also use this code to unlock various free classes on Unacademy. Again, the link is in the description box. Click, subscribe and share. Don't forget to use the referral code VinAmG. Now let's get into the video. Now let's see the first question, which is from lac operon. The lac operon in E. coli is controlled by both the lac repressor and the catabolite activator protein, CAP. In an in vivo experiment with lac operon, the following observations were made. A. CAMP levels were high. B. Repressor is bound with allolactose. C. Catabolite activator protein is interacting with RNA polymerase. Which one of the following conclusions is most appropriate based on the above observations? First, the glucose and lactose are present. Second, glucose is present and lactose is absent. 3, both are absent. 4, glucose is absent and lactose is present. Now let's look at how you're going to solve this question. So now let's look at the first observation, which says CAMP levels are high. Now CAMP level in any cell, it is dependent on the level of glucose. So if the glucose level is high, then CAMP level is low. And if the glucose level is low, the CAMP level are high. Now this is because when glucose is present, it basically prevents the activation of the protein or the enzyme which is required for the conversion of ATP into CAMP. Okay, so if a cell contains high level of CAMP, that means glucose in that cell is absent. So from first observation we get that glucose is absent. Now let's look at the second observation, which says repressor is bound to allolactose. Now if repressor, when repressor is produced in lac operon, it is present in the active form. Now when allolactose bind to the repressor, it becomes inactive. Now the presence of allolactose in the cell means that lactose in the cell is also present, because allolactose is an isomer of lactose. So when lactose enters the cell, it is converted into allolactose, which acts as the inducer in case of lac operon. So the presence of allolactose in cell means the lactose is present. Now let's look at the final observation, which says CAP is interacting with RNA polymerase. Now CAP protein interacts with RNA polymerase only in the form of CAP-CAMP complex, which again goes back to that CAMP is present in the cell, which means glucose is absent in the cell. So from these 3 observations, we can conclude 2 things, that glucose is absent in the cell and lactose is present in the cell, which gives us the answer to this question. That is the 4th option, that glucose in the cell is absent and lactose is present. Now let's look at our next question, which says what will be the, with reference to lac operon, what will be the phenotype of an E. coli strain with genotype I minus, O plus, Z plus, Y minus, and on F plasmid I plus, OC, Z minus and Y Y plus. And our options are constitutive for both beta galactosidase and lac permease, inducible for both, that means you require an inducer for the expression of these two genes and for the production of beta galactosidase and lac permease enzyme, and the inducer in this case is allolactose. Now the third one says inducible for beta galactosidase and constitutive for lac permease, and the fourth one is constitutive for beta galactosidase and inducible for lac permease. So now let's see. This E. coli cell basically contains two copies of lac operon, one on the chromosome and another one on the F plasmid. Now on the chromosome, the I gene is I negative, which means it is mutated. It is a defective gene. It either does not produce a repressor or it produces a defective repressor. Okay. And O positive, that means this gene is just fine. Now I gene produces a repressor protein. So I gene produces a protein that can move to other locations and bind to other locations, which means that the I gene is a transgene. And the protein produced by it, that is a repressor protein, is a trans protein. Now the Z and Y, these are the structural genes. Z encode beta galactosidase and Y encode lac permease. Now let's look at the F plasmid. The I gene is I positive, which means it can produce an active repressor. Now OC is a defective gene, so the repressor cannot bind to this OC. However, it can bind to this one. What happens when the lactose is absent? Now in case lactose is absent, this repressor produced by I plus gene will bind to this gene, this operator. It cannot bind to this operator because this operator lacks a binding site for the repressor. However, it can bind to this repressor. So the repressor will bind to this gene, and since the repressor is bound to the operator, these two genes will not be expressed. Okay. So that is the first condition. So these genes will not be expressed in the absence of lactose. However, in this case, since the operator is defective, even if the lactose is absent and the repressor is produced, it cannot bind to this operator. So these two genes will be expressed at all the times, whether lactose is present or absent. So these genes will be expressed at all the times, and these two genes depends on the presence of allolactose to bind to the repressor, and so the repressor becomes inactive and cannot bind to this operator. So this part is inducible and this one is constitutive. So since this is constitutive but the Z gene is negative, it cannot produce an active beta galactosidase, so it doesn't matter. However, Y gene can produce lac permease, so the lac permease will be produced at all the time, which means the Y gene will be expressed at all the times, which means the Y gene is constitutive. Okay, so in case of phenotype, the lac permease will be constitutive for lac permease. Now let's look at this part. Since these two genes will only be expressed in the presence of lactose, which means the Z gene is inducible, so the lac operon is inducible for beta galactosidase. So let's look at our options. Constitutive for both, no. Inducible for both, no. Inducible for beta galactosidase and constitutive for lac permease. So that's our answer, as according to this one, it is inducible for beta galactosidase, and according to this one, it is constitutive for lac permease. Now let's look at our next question, which says mutants of lac Y gene, that is Y minus, of E. coli do not synthesize the lactose permease protein. The following statements refer to the behavior of lac Y minus mutants under different experimental conditions. So let's look at our options first. The first statement says no synthesis of beta galactosidase when Y cells are induced with lactose. Y minus cells are induced with lactose. Now this statement is true because even though the lactose is present in the medium, the entry of lactose into the cell is only done when lactose permease is present. It mediates the entry of lactose into the cell. So even though the lactose is present in the medium, there will be no synthesis of beta galactosidase because lactose won't be converted into allolactose and the genes won't be expressed as the repressor will be still bound to the operator. Now let's look at the second option, which says synthesis of beta galactosidase when cells are induced with lactose. As the first statement is true, the second one cannot be true as they are just opposite of each other. So the second statement is not true. So A is true, B is false. Now let's look at the C option, which says no synthesis of beta galactosidase when cells are induced with IPTG. This statement is false because IPTG can enter the cell through other mechanisms also. It does not just require the presence of lactose permease enzyme. So IPTG can enter the cell through another mechanism, and this can induce the lac operon and beta galactosidase can be synthesized. So the C is false. Now let's look at the D option. Synthesis of beta galactosidase when cells are induced with IPTG. Now this is true because this is just the opposite of C option. In the presence of IPTG, the lac operon will be induced and beta galactosidase enzyme will be synthesized. Now let's look at the E option. The cells induced with IPTG cannot grow in the presence of tone PG. Now this is false because the tone PG is a compound whose uptake is mediated by lactose permease. Now since lactose permease enzyme cannot be synthesized and it is not present in the cell, tone PG cannot enter the cell, it will not be taken up by the cell, and so the toxic compound will not be produced and cells will be able to grow in the presence of tone PG. So E statement is false. So let's look at the F one. Cells induced with IPTG can grow in the presence of tone PG. Now this is true. This is exactly the opposite of E statement. So we have A is true, we have D is true, and we have F is true, and rest of the three options are false. So our answer for this question is the first option, which says the A, D and F are true. Now let's look at our next question, which says attenuation is a mechanism involved in the regulation of tryptophan operon in E. coli. Now when tryptophan levels are high in the cell, region 2 of the TRPL is blocked from pairing with region 3. This allows the pairing of region 3 and 4, leading to the formation of a row-independent terminator. What would be the structure of the TRPL region in E. coli cells when protein synthesis has been inhibited? Now let's look at the first option. Region 2 and 3 will pair, allowing the transcription of structural gene. Now if you have gone through the TRP operon properly and you've seen the mechanism of attenuation, you know that region 2 and 3 will only pair when region 1 is blocked for some reason. Now in this case, since protein synthesis is inhibited, after transcription, translation does not follow. So there is no ribosome blocking any of the leader sequence. The leader sequence consists of 4 regions, 1, 2, 3 and 4. 1 can base pair with 2, 2 can base pair with 3, 3 can base pair with 4. So 2 has the option of base pairing with 2 regions, 1 and 3. Now in this case, since translation is not happening, there is no ribosome blocking the region 1. So when transcription of region 2 is done and the RNA polymerase move to the region 3, region 2 is free to base pair with region 1, which is not being blocked by ribosome. So in the absence of protein synthesis, region 1 and 2 will base pair, which leaves region 3 and 4 to base pair with each other. And as we know that the base pairing of region 3 and 4 results in the formation of a hairpin structure, which is also known as the loop terminator, which leads to the termination of transcription. So the structural genes are not expressed. So the second option, which says R1 and R2 will pair, leaving R3 and R4 to pair, resulting in the attenuation due to the formation of a terminator loop. So this option is the correct answer. Now let's look at our next question, which is a merodiploid strain of E. coli with the genotype F plus OC Z negative Y plus oblique O plus Z plus Y plus was constructed. The activity of beta galactosidase enzyme was measured in this strain upon following treatments. First, let's look at what will happen under each of these conditions. So under no induction, see, even if there is no lactose or any other kind of inducer is present, a small amount of enzymes are always produced. So gene expression on a small level is always happening even in the absence of any kind of inducer. Now this is required because when lactose is introduced into the medium, there are certain enzymes that will be required. For example, beta galactosidase will be required for conversion of lactose into allolactose that acts as the inducer. Lac permease will be required for the uptake of lactose. So a small amount of these enzymes are always being synthesized even in the absence of induction. Using this part, we can actually eliminate the first and second options of your answer. This is because in first option, if you look, the A, B, C have the same level of enzyme activity, which is not possible because the level of expression of these genes is going to be different under these conditions. As under no induction, only a small amount of enzymes will be produced, only a small level of gene expression is going to happen. So we can eliminate the first option. Now we can also eliminate the B option, the second option, as it says that in case of no induction, there will be no enzyme produced. There will be no beta galactosidase enzyme produced, which is not possible, as we just discussed that even in the absence of any inducer, a small amount of enzyme is always being produced. Now if you look at the third and fourth option, for A, which is no induction, it shows that a small amount of enzyme is being produced. Now look at for the B and C one. Now in case of 3, it shows that the enzyme produced on induction of IPTG, which is the B option, is more than the enzyme produced on induction with lactose. Now this is actually true. This is because when lactose is introduced into the system, it does not only act as the inducer, it is also involved in other metabolic pathways. However, IPTG is not involved in any of the metabolic pathway, so it acts as a better inducer for lac operon, and the expression of lac operon is better when IPTG is acting as an inducer. That's why the enzyme produced when IPTG act as the inducer is more than the enzyme produced when lactose is acting as the inducer or allolactose is acting as the inducer. Now to confirm that third is our answer, let's look at the graph for D. Induction with n moles of lactose in the presence of n moles of glucose. Now for third, it says that a small amount of enzyme will be produced. However, for 4, it says that 0 amount of enzyme will be produced. Now the third answer is correct because even when the glucose is present and lactose is present, both lactose and glucose is present, although the expression of lac operon is less as compared to in the absence of glucose, but there is expression of lac operon, so some amount of enzymes are being synthesized even when glucose is present in the system. So this concludes that the answer for this question is 3. Now these were the 5 most important questions that you can expect in your exams, or not as such, but you can expect questions related to these kind of questions. Now VinAm Guidance is also going to organize a quiz on fundamental processes. Now this quiz will include questions from replication, transcription, translation, regulation of gene expression, Operon system including lac operon, mutation in lac operon, TRP operon, ARA operon, and we'll share the link for the quiz soon on our YouTube channel, and I request you all to please attend this quiz as it will help you monitor your progress for these topics, and also it's free. So please attend this quiz and please stay tuned for more updates on the quiz and other events. So that's all in this video. I'll see you in the next video. Please don't forget to subscribe to this channel and hit the bell icon for latest updates. Thank you.